Becca Reef joined the APBD Research Foundation team this past summer after earning her Master's of Science in Genetic Counseling from Penn. Becca is committed to drive progress in rare disease research and patient advocacy to support the development of new initiatives aimed at empowering patients, fostering collaborations, and accelerating drug development. We're excited to have Becca with us, and now I'll turn it over to Deborah Goldman.
Hi, everyone. Thank you for joining tonight. We have a fabulous guest speaker, Becca, and she will talk to us about biomarkers. And as Calliope just said, we'll leave our questions till the end, but feel free to put questions in the chat and we have time to do everything. Okay, go ahead.
Thank you so much, Deborah and Calliope. And hi, everyone. If we haven't had the pleasure of meeting one-on-one or in a group session, my name is Becca. I joined the team in August, and upon some discussion with our board team, we decided that based on some feedback from our conference, such as that the event was just so chock full of information that some of it was difficult to digest and some of it was very scientifically heavy, using a lot of specific terminology that could have been difficult to understand, especially in the context of 6 hours of presentations. We decided that it would be a good idea just to break down some of the advances as well as topics that were presented at the conference. So I wanted to start today with biomarkers. So this will be about a 10 to 15 minute presentation on what biomarkers are and some of the important biomarkers for APBD in particular. I tried to simplify it as much as possible. So we'll just go through this, and if anyone has any questions, you can feel free to type them in the chat throughout, or we can go over them at the end. And if nobody has any questions, I have kind of a list of just thought-provoking considerations for us. So with that, I'll just get started. You may remember from our conference, we had 2 sessions on biomarkers. We had a session outlining the critical importance of them and the need for them to move forward with clinical trials, as well as a session on specifically biomarkers that are in development for APBD. And so the outline of this presentation today will just be a quick overview in the simplest terms of what biomarkers are. We'll go over the 6 key types of biomarkers, biomarkers in development for APBD, and then what this means for us as a patient community. So what are biomarkers? I sat down and I really tried to think of the simplest possible definition for a biomarker. And what came to me is that a biomarker is a signal from your body, just telling us what's happening inside. And so we're searching for a bunch of different signals that can either tell us a bunch of different things or individual signals that are telling us something really specific about what is going on inside. So the definition, according to the FDA and NIH working group, is a little bit more complex than that. I thought I'd throw it up here just to be a little bit more specific, but they define it as a characteristic that is measured as an indicator of normally what's going on inside your body or a response to an intervention such as a medication. And so why are biomarkers important? They help to confirm the presence of a disease. They can also help diagnose diseases earlier. And most importantly for us in the context of treatments, they can monitor treatment effectiveness and safety. And nowadays, when the FDA looks to approve certain drugs, treatments, medicines, clinical trials, they really look for work on biomarkers because they can help not only our patients stay safe, but also really give us a lot of information on whether an experimental treatment is working. Now, there are a ton of different types of biomarkers. You may remember Dr. Nadowitz at our last conference talking about a bunch of different types, and he went into quite a bit of detail. So I'm going to outline the 6 types that he talked about, again, in the simplest form. And again, I just want to reiterate that there's absolutely no need to remember all of these different types of biomarkers. If any of you are specifically interested in biomarkers that are relevant to APBD, we'll go over those in just a moment. So there are 6 important biomarkers in terms of categories. There's diagnostic, monitoring, predictive, risk, safety, and therapeutic. And we can take a bit of a deeper dive into each one. So a diagnostic biomarker may be exactly as it sounds. It helps to confirm the presence of a disease. So what's an example of this? A diagnostic marker can tell us whether someone has APBD. So an example of this in practice would be something like if you get a genetic test that tells you that you have a mutation in the gene that leads to symptoms of APBD, or gives you the diagnosis of APBD. The second type is a monitoring biomarker. This type of biomarker, again, is something inside your body that we can measure that tracks the progress of a disease or a treatment response. So why does this matter? It helps us to see if any treatments are making a difference. So for example, you may try a new treatment that becomes available in a few years. And if your healthcare team wants to see if it's working, they may be able to measure some levels of certain molecules in your body that will tell us, yes, this treatment is working, or maybe no, let's switch gears to something different. A predictive biomarker indicates how you may respond to a treatment. So this is something that could indicate an increased likelihood of a response to treatment and can help us choose treatment. Now, this type of biomarker may be a bit further in the future for APBD, but it could be something that could be important for us, in the future when hopefully we have multiple different treatment options to choose from, and we may be able to see a signal in the body that's telling us that something might be better than another thing for a specific person. We have risk biomarkers, which can show the likelihood of developing a disease or a condition. So an example of this would be something that helps us identify who might develop APBD or be at increased risk for a certain clinical change, such as bladder dysfunction. So this would be a signal in the body that tells us, maybe this person is at an increased risk than the person walking on the street to develop a symptom because we're seeing this level rise over time. Second to last, we have safety biomarkers, which detect potential harmful effects of a treatment. The application of this is that they help protect our patients during treatment. An example of this in practice would be doing something like taking a complete blood count or doing liver function tests to ensure that a treatment isn't producing harmful or toxic effects on the body. Our last type of biomarker is a therapeutic biomarker. These indicate the effectiveness of a specific treatment. Is it doing its job? This would be a signal in the body that we can measure, such as the amount of accumulation of glycogen. If that is decreasing, then it seems like whatever therapy we may have will be doing its job. And so that is a general overview of the 6 different types of biomarkers. And now we can jump into what kinds of biomarkers are currently in development for APBD. And just before I jump in, to clarify, although there are 6 distinct types of biomarkers, they can also overlap. So for example, we may have a biomarker that will tell us if somebody is at an increased risk for developing a certain change, but it also may be able to be used as a therapeutic biomarker. The level of that molecule in the body may go down with the treatment. So now I will present the biomarkers that were discussed at our conference. And again, I can make these slides available for dissemination, so don't feel the need to take any notes or write anything down unless that's something that's helpful for you. So we can collect biomarkers in different ways. Again, these are molecules or signals in our body that are measurable, so they can give us information. One type of biomarker is a biomarker collected from a urine sample. And at our conference, we heard from Dr. Mayank Verma at UT Southwestern about a biomarker that was actually hinted at because the Pompe disease community is already using this biomarker. The biomarker is called GLC4. That's less important than the concept as a whole, but it's a molecule found in urine, and it helps diagnose Pompe disease, which is a glycogen storage disease 2, while we're a glycogen storage disease 4. Now, Dr. Verma's research shows that these levels are also higher in urine samples from APBD patients, which suggests that this could be helpful. This could potentially, in the future, be something that we could also use as a diagnostic biomarker, like our peers in the Pompe community. We also have biomarkers that can be found in the blood. One biomarker that has been found in the blood can be a sign of brain health. This biomarker is called GFAP. Again, name isn't super important, but essentially this is a molecule known by scientists and researchers to be found in the blood when there's brain injury or disease. Dr. Verma from UT Southwestern found that the levels of this molecule are higher even in carriers of 2 different mutations in the gene that causes APBD, as well as in mouse models of APBD, which may help us better understand the disease. An example of how this could be utilized for further understanding the disease is that we can try to identify at what age this biomarker starts to become elevated, and could this potentially help us diagnose APBD earlier, especially because many of our community members go through misdiagnosis. There are really kind of a myriad of ways that this could be utilized. Another biomarker in the blood is something called neurofilament light chain, abbreviated as NFL. This is a molecule that has a lot of applicability in other neurodegenerative diseases. It's already used as a biomarker in diseases like ALS, as a diagnostic biomarker, as well as a monitoring biomarker and a therapeutic biomarker. It tells us if somebody has the condition, it tells us if it's progressing, and it tells us if treatment is working. Dr. David Brunner from Ulm University presented at the conference that neurofilament light chain is also higher in Lafora disease patients, which is one of our allied diseases, and correlates with how long they've had the diseases and their symptoms. So patients with Lafora disease, the patients that have had it the longest, have higher levels of this biomarker. The patients that have increased severity scores based on their performance scale have an increased number of this biomarker. And why that's relevant to us is because there are a lot of similarities on a molecular level between Lafora disease and APBD, and this could also potentially be used to monitor APBD, which is something that the GHF group in Israel is also working on. And we will do a whole separate patient chat presentation on that therapy. Another type of biomarker is one that we can see through imaging, such as MRI. Dr. Nurbeh Yadav from Johns Hopkins is developing a new type of MRI that can detect changes in glycogen in the brain more effectively. Early studies show that this method can reveal differences in glycogen before and after treatment, which is really helpful to us as we try to move forward with having a tool in our pocket for if and when treatments become available, to be able to show that the treatment is working. Because as I said, this is very important to the FDA when they are looking to approve different treatments. And so you may be asking, this is all very exciting work. It's good to hear that there are potential biomarkers on the horizon that can either diagnose APBD earlier, monitor the progression of APBD, or even tell us if potential future treatments are working. And so what does the ongoing research and development look like, and how does that affect us? And so right now, all of the biomarkers that I just described are in various stages. Some of them have been tested in mouse models. Some of them have been tested in human samples. That we're in stages 1 and 2. So what are the next steps? The next steps would be validating these measures, which means making sure that we can ensure clinical validity, which means that no matter what hospital you're getting these biomarkers collected at, whether that's via urine, blood, or imaging, it's standardized across all centers. We're making sure that we are measuring the same types of measurements. We want to use the correct statistical methods. We want to ensure that the false positive rate isn't something crazy. And then once we have all of the analytical and clinical validation done, is this biomarker something that's acceptable? Is it something that's cost prohibitive? Is it something that our partners at different academic institutions have the technology to use, and such? Now, how can you get involved with biomarkers at this stage? So as I mentioned, we have different studies in different phases, phase 1 and phase 2. Excitingly, we have 2 studies that are really entering phase 2 so that we can progress to phase 3 of validation. That first study is the P for APBD urine biomarkers study. Dr. Minassian and Dr. Verma at UT Southwestern are looking to recruit a number of patients, either patients with APBD, individuals with the diagnosis but no symptoms, carriers, or family members that do not have the mutation, to provide a urine sample. And I will provide more information about this in the chat if you are interested, and interested in moving this research forward. We also have an innovative MRI biomarker study, which is a bit more difficult to participate in, to be frank, but still something that is available to you all. Dr. Yadav is looking to recruit 5 to 15 patients with APBD who are able to travel to Baltimore, which we very much understand that not everybody is, to undergo MRI scans. Hopefully one day we will hope that this technology will be available at other medical centers, but for right now, it is unfortunately only available at Johns Hopkins, which is why he is asking patients to travel to Baltimore. We know that that is very difficult, but if anybody is in the general vicinity or area and is interested in traveling, then you do have the opportunity to do this scan and contribute to the research. And that is pretty much everything. So I will stop sharing my screen for now, see if anybody has any questions. I can always reshare my screen, bring up certain slides again. And if nobody has any questions, then I can provide some of my own thought questions for all of you. Thank you so much.
Thank you so much, Becca. Let's see, Deborah, I know you had a question.
Okay, this is actually a question that someone wrote into the chat prior to this meeting, but thank you very much, Becca. That was wonderful. What are the difference between clinical endpoints and biomarkers?
That is an amazing question and gets a little complicated because it's a concept that's not exactly mutually exclusive. So while they kind of are their own two definitions, you also could have a biomarker that is an endpoint. And so when people talk about clinical endpoints, they're talking about a measurable outcome to a treatment that may be through a clinical trial. So an example of this would be, just giving kind of a lay example. If you have a fever and take Tylenol and your fever comes down, that is a measurable outcome of the Tylenol working. And so in our context, we may say, in 2 years, 3 years, 5 years, if we have a treatment, we have to start to think about what kinds of outcomes are looking for. And, that's something that's important to us from a patient advocacy perspective, to ensure that any pharmaceutical company that's interested in developing a treatment is developing a treatment for what our patients' concerns are. Because sometimes a pharmaceutical company can come in and think, okay, well, what we want our endpoint to be is reducing the percentage of glycogen buildup. But if that doesn't actually improve any symptoms that our patients experience, we may want another endpoint that's actually, no, we want our endpoint to be more subjective. We want the endpoint to be that people have increased mobility on the scale of 3 to 5 years from what they typically have. And in that same vein, we could also say, hey, we know that this specific biomarker predicts our symptoms. So we could also have an endpoint that's our endpoint of this trial or this treatment is that this biomarker lowers by this certain percentage.
No,
Does that
that's
make
great.
sense?
Total sense. That's very clarifying.
Great. I believe, Larry, I saw you go off mute if you have a question.
Oh, I just went off mute, but I was sort of thinking of, so Dr. Verma is working on biomarker in the urine, which I'm assuming at this point, if he gets enough samples, he may find an increased level of something in the urine. The next step, though, is to try a proposed therapy, I take it, to see whether it has any influence on that biomarker. Are there any therapies in the pipeline that might then be able to be applied using the biomarker?
Yeah, that's a great question. So to be candid, there is nothing available or that is going to be imminently available at this moment that we know will have an impact on any specific biomarker. The goal of advancing the biomarker study is that so when that treatment or drug does come down the pipeline, we are ready to move forward, and there will be nothing standing in the way of moving forward with clinical trials, getting that treatment to our patients. Does that answer your question, Larry?
Yes,
Okay,
thank you.
perfect.
Great, thank you, Larry. Becca, did we get Howard's question in the chat also?
Yes. I can read it out loud if that would be helpful.
Yes.
Great. So Howard asked that currently some of the studies looking for biomarkers involve an MRI, which is Dr. Yadav's study. But unfortunately, there is some exclusion criteria. So if someone has something like a cardiac stent, then they are not able to have an MRI for safety reasons. And so this may lead to disproportionate access. So if these markers help find a cure, then would that patient not be eligible for that treatment? That is a great question. Theoretically, if an MRI was the only methodology that we had to be able to say whether a treatment was working, I could see that being a barrier. I don't have enough knowledge of MRI to be able to make a statement as to whether there are exceptions to that rule. That is definitely something that I can speak with Dr. Yadav about and get back to you all on. I would think it would be unlikely that we would be in a situation that you would have to get an MRI to be able to be eligible for treatment. The purpose of the MRI could be to assess if the treatment is working, but we are also hoping to have other markers that can also give us that information. So hopefully we would not be in a situation where not being able to get an MRI would be exclusionary from any potential treatment. But again, I will check in with Dr. Yadav and can bring that information to the next patient chat event or email anybody that's interested directly.
And I believe that is true for any of the compounds or potential treatments that are being looked at. It will not be assessed by just one measure.
Mm-hmm.
Great. We had a couple more questions come in to the chat. Are biomarkers being used for GHF1?
Yes, they are. And I'm hoping to have another patient chat event dedicated to all of the work that GHF is doing over in Israel on their compassionate use trial and study, because they presented a lot of information at the conference. There were some connectivity issues. Sometimes the videos weren't on. So I think it would be great to just debrief everything that was said.
That would be great. Yeah.
But in general, yes, biomarkers are being used. Their group is not developing any new biomarkers, but they are using all of these same biomarkers that I just described, including the neurofilament light chain and the GFAP, to see if they are seeing any measurable difference in the 3 patients that they are treating. And there are results on that they presented at the conference. They do see a decrease in these biomarkers, which is a good thing. The only trouble there is that that's not data that, our healthcare system or FDA accepts when they are making decisions on treatment efficacy, but it's definitely a good sign and moving in the right direction. And to answer Kathy's question as well, are they working on biomarkers? So as I had just said, I realized I left out a small detail. The GHF group is not specifically working on developing new biomarkers. They're using these same biomarkers that I just described to assess if their treatment that they've developed is working. Now, Dr. Caclone, who works with the GHF group, but is also his own independent researcher, is working on potential new biomarkers. And that is a project that was funded by our $1 Million Bike Ride grant. The results of that are not yet out, but when they are, we can definitely, be transparent and discuss the results of this potential new biomarker that he has created.
Great, thank you. And I know we had some questions in the audience from Karen.
Okay. I was wondering, since we live all over the place, all over the US, I have a neurologist who is very interested in rare diseases. And she's, up at National Jewish Hospital and state research facility. And I'm wondering if, since travel is an impedance to like come to Baltimore and, just from what it takes to travel, if there could be any partnerships. I mean, if are they open to having some kind of partnership to try and get the information you want from an MRI? But
Mhm.
so you got willing participants, but
Mhm. Mhm.
travel makes that more difficult.
Mhm.
What do you think? Or is that something that you could check with?
Yes, I absolutely hear you. It is very frustrating to have this kind of research available, and then like the one reason that you can't participate in it, even if you want to, is travel. The reason why it is currently limited to one site is because it's a very new technology that I was going to say not all, but I would say most sites actually don't currently have the bandwidth to support. But I will say I know that Dr. Yadav is working on a solution to this problem. He is working on applying to different grants that may be able to give the funding for other institutions to participate in this research and develop this new technology on their own. It is something that is in the works. They are trying to make it more available. And again, as I had said previously, and Deborah had also mentioned, we really don't think that if while it is extremely frustrating to not be able to participate in something that you're interested in, participating in this would not then exclude, or not participating in this wouldn't then exclude you from if there was anything that came out of this study. But yes, to answer your question more directly, they are working on making it available. Do I think it will be available soon? Probably not. Probably closer to the scale of 1 to 2 years, if I had to give more of like an accurate timeline. And I also wanted to add on top of that, Karen, that I actually met with the two co-chairs of our scientific and medical advisory board today. And we developed a plan moving forward to ensure that the coordination of care of all of our patients at centers across the United States is more organized. So we're going to be convening a group of clinicians from across the country and developing kind of a standardized spreadsheet on what they should be collecting at each visit. So if this is something that any of you think would be of interest to your own neurologist, urologist, any of your providers, and you would like them to be a part of that conversation, you can feel free to either email me with their information or put it in this chat, and we can ensure that they are involved in all of those conversations as well. That is separate from the MRI because that is just a technology issue, but that we're hoping that that will help ensure that there's continuity of care and all of our patients are receiving equitable care and receiving the top of the line options available.
Great. I was just thinking that collaborative piece in terms of, as you said, other institutions do have money, maybe. Or maybe there's money from the $1 million grant that could be, whatever would be needed to get more information from people.
I have another question in the chat on whether there is any progress on making the treatment that's being used on 3 patients in Israel available to patients in the United States. And I wanted to address that. I wanted to just pull something up here on my computer just to make sure I'm giving the correct information. I believe that the timeline that was given by GHF at our conference, oh yes, here it is. The timeline that was given by GHF at our conference was going to be entering the next phase in their drug development by the end of 2025, and then filing to get approval for the drug closer to 2027, 2028, which I know is not the best answer. I wish I could give a better answer because that is still 3 to 4 years away. But that is their current timeline. Their current timeline is sub 5 years, which I think is good news. And if that is expedited in any way, we will be transparent and you will all be the first to know.
Great. We have another question that just popped in the chat.
So Kathy spoke to Dr. Schiller in Texas, and he told her they were working on an MRI that would diagnose APBD in an MRI. Is this the same as the MRI in Baltimore? I believe this is the same MRI as the one in Baltimore. I could be wrong on that, but I do know that the site at UT Southwestern is one of the hospitals that Dr. Yadav is working with to try to work on a grant to expand this MRI technology to that institution. So I do believe that this would be the same MRI. It is possible, though, that it's a different MRI because APBD can be diagnosed by a standard conventional MRI. Yeah, these have been great questions so far. I know that it's difficult to, hear this information that's very exciting and then feel like we're kind of playing this waiting game when, we don't quite know when these things will be available. So I think you all are doing such a great job in asking all the right questions in trying to push this forward.
Great. Thank you so much, Becca. We have another one from Mark in the chat.
And Mark says, oh, I lost my screen.
So
Let me
what
get it
size
back.
sample is needed to validate a biomarker, given this is a small community? Are there enough volunteers to get a conclusion?
That is a great question. And luckily, that is not something that's set in stone. So just because we have a small community doesn't mean that we won't be able to validate it. Now, it will depend on how reliable that biomarker is. So for example, if we have a biomarker that has a really obvious big effect, for any of you more scientifically minded people, this would be a biomarker that has a huge statistical significance, we would need less people to validate it. And also if the biomarker measurements across institutions that were collecting them were very similar, we would need less people to validate it. Now, if the effect of the biomarker was quite small and it wasn't as reliable in diagnosing APBD or telling us if a treatment was helping APBD get better, or if there was quite a range in variability amongst different patients that had different measurements taken at different institutions, we might need a larger sample size of patients. So unfortunately, the answer is that I don't know because it will be quite subjective. But the good news is that it's not like we need 500. We could just have 10 if it's a strong enough biomarker.
Great. We also had Larry with another question.
No, sorry.
Oh, okay. Just going off mute. That's okay. No problem. Anyone else? Feel free to post your question in the chat, or if you go off mute, I'll also see you pop up on the screen and I can call on you that way.
Another piece that I can share too is that I was at a few different conferences throughout the month of October trying to learn from allied rare disease groups or peers and other rare disease groups to really try and understand what the hurdles are in getting us to move forward. And one piece of information that I did learn is that it's very important to the FDA when they're making decisions on whether to fund or approve treatments to have a formal meeting with them to address what we know, what we don't know, what we're trying to develop, what's in development. And so right now, the foundation is discussing what that next step, a formal meeting with the FDA over the next year, end of 2025, beginning of 2026 could look like to start to address some of these hurdles and start to be able to give more transparent answers on why these things are not currently available and what it would take to make these types of measurements available to you in clinic and then available if and when we have a treatment.
Thanks, Becca. I saw Mark go off mute. Did you have a question, Mark?
Yes, I was under the impression that GHF1 was approved for compassionate use in the United States. Is that happening, or is it going to happen?
I am less familiar with that, Deborah. Do you have any insight that I don't?
It is not my understanding that it has been approved in the United States. They went to the FDA. They've gone through the first stage of assessment with the FDA, and that's all that's happened. The only place it's approved for is only 3 patients in Israel. Our hope is that it will at some point be approved in the United States, but that has not happened yet.
And again, the timeline that they had given us was between 2027 and 2028 for when they
Yes.
envisioned
Yes.
FDA approval.
Yeah.
Thank you. Aura, I see you went off mute. I didn't know if you had a question.
Yeah, I was just wondering whether that was the FDA as part of an FDA breakthrough device. Is that the pathway, or there's a separate pathway for ultra rare diseases because the clinical trials are just different in terms of their design? Do you know what that, what either for, Golden Heart or for, the ASO therapies or anything else that's coming in, and the MRI device related studies? I mean, what
Yeah, so I know that Golden Heart Flower is aiming to take
can
advantage of.
to-
Oh, sorry, I might have accidentally interrupted you, Aura.
No, you're
No,
fine.
no, go ahead.
Oh,
My
okay, perfect.
connectivity is not that great. Yeah.
That's okay. So I know that Golden Heart Flower is aiming to take advantage of a program through the FDA called the Orphan Drug Designation Program, that will hopefully accelerate the timeline. And, I know it feels kind of dreadful right now to look towards 2027 and 2028, but 2027 and 2028 would be that expedited timeline under the orphan drug designation. They presented on this a little bit at the conference, just kind of describing that there are certain tax incentives, certain financial incentives, such as vouchers for commercially making the drug available, that make this an attractive pathway to pursue. And it also decreases the approval time from 12 to 15 years to 8 to 11 years. So it does give us a bit more, a bit less waiting, but that still does take us to 2027, 2028. In terms of any other therapies that we are pursuing, some of you may have heard that there is a drug for the Lafora disease community, and Dr. Manassian had mentioned at our conference that theoretically proof of concept is there, that it could work in APBD, but we don't have a pathway to use it. That is something that we're exploring. That would be something that could be considered a basket trial, which is something that actually is harder to get approved by the FDA because you're treating 2 different conditions, especially because that condition is pediatric and our condition is adult onset. So that would be kind of its own pathway as well. In terms of the ASOs are unique because right now, the ASO therapy that we are thinking about is specifically for patients with an intronic mutation. And ASOs are a technology that has already been FDA approved for its safety. And so they're able to be produced and given to our patients on a much faster timeline because it's already been proven that they're safe and efficacious. So I think those are the big 3. I'm trying to think of any others that could have kind of their own pathway, and nothing's coming to mind. I'm happy to do a little bit more work looking into if there are any other specific pathways that we could pursue. But I can send you, Aura, the slides from GHF that go over how they utilize the orphan drug designation, if that would be helpful.
Sure, that would be great. And I think it's probably on the symposium on the
On the website.
recorded. Yeah, that I can go back and review. I just didn't.
Yeah, no problem. Their website seems like it's from 2006. It is very difficult to navigate. So I can send it to you. It's no problem.
Great, thank you. Kathy, it looks like you went off mute. Did you have a question?
Yes, hi, thank you for being here, everybody.
Fine.
Before I found out I had APBD, a lot of us have had this experience. I was diagnosed with all sorts of diseases and went to all sorts of support groups, trying to find out what's wrong with me. And for several years, they thought I had CMT, Charcot-Marie Tooth, which is a very old muscular dystrophy disease. So CMDA, and then then I found out after tons of genetic testing that I had APBD, and indeed my brother as well. And the thing about what I'm questioning is I still get paperwork. I get their Quest or whatever it's called, their magazine, and they were talking about having a cure or treatment, not a cure, for CMT. And we have a lot of the same symptoms, not all. And I was wondering if that had been discussed, the crossover between muscular dystrophy like CMT being so much like APBD. If that would warrant. And another question is the 3 patients that they're working with in Israel right now. There was a period when there was a lot of us that were going to go over there for the treatment, and then the war started. Well, COVID started, and then of course the war. So I didn't know if that was still open or if that was a closed deal now.
Yeah, so I think that is a great thought process that, given the similarity in treatment for CMT, why isn't it available for us? So I will say just off the bat, if the treatments that are available for CMT are symptomatic, such as treatments to help alleviate neuropathy, I see no reason why you couldn't ask your healthcare provider if you were eligible for that treatment. If it was more of, like a cure or gene therapy, the reason that we may not be eligible for it is because the gene is different, as you know. And the reason why we think of ourselves as kind of allied with other diseases such as Lafora disease, but maybe not diseases such as CMT, are that Lafora disease is also due to a mutation in a different gene, but it interacts very, very closely with the gene that is mutated in APBD. So the thought process is that if one of them is fixed, the other will be helped. And so that's why we think about those two kind of closely together and think that, an advance in one community is an advance in the other, whereas CMT, the gene that is mutated and that they're looking to target, unfortunately does not interact with the gene that we have mutated in APBD. So it wouldn't work as well for us. Now, in terms of the golden heart flower availability, I was under the impression that unfortunately they were only authorized to treat 3 patients, and they have selected those 3 patients. The concept of traveling to be treated was before my time, unfortunately, with the organization. So, Deborah, I'm not sure if you have more insight than I do.
Well, I think that initially before they only got approval for 3 patients. So until they get further approval in the United States, that is not an option for us. And it also is still in the research phase of, they believe this is helpful, but it hasn't been validated. They're not saying this absolutely is a treatment and we want to go ahead with it at this dose for this amount of time. They're still working all those details out, which is why they said 2027, because no one wants to be given a medication that they're not sure works and has efficacy, because it's timely and costly. So they're doing the work. Not the best news, but I think that is the reality.
Thank you.
And the other thing is that any new treatment that we hear of, whether it be at a pharmaceutical company in a foreign country, anyone that is doing any work on glycogen storage disease or allied illnesses, we explore our options because we desperately want to find some treatments and some cures ultimately. So it's an active, ongoing search always.
Yeah, and I definitely want to echo that, Deborah. I know that unfortunately, especially given kind of the constraints on our healthcare system, many of you are used to the feeling of feeling not heard, ignored by your providers, like you had nobody to advocate for you. And I just want to reassure all of you that we are addressing every potential avenue here at APBD-RF. You know, we're talking as a group at least once a week, usually every day, about new developments, what other groups are doing, any potential. And you can always feel free to reach out to any of us to ask, you know, things like, oh, this CMT group is doing this. Like, what's up with that? Why can't we participate in that? Always reach out, but also if it even helps you get 5 more minutes of sleep at night, knowing that we are doing the work every single day to explore every potential avenue, as Deborah said, I hope that it at least brings you a little bit of peace.
Thank you so
Of
much, Becca.
course.
This was very clarifying and really, really helpful.